Good day everyone, I am Pauline Langido and today I will impart about the cell motility, the integrating rule of the plasma membrane. Now I will be going to tackle about the membrane curvature, the membrane transport and flow. What is membrane curvature? It is a motile cell contains regions characterized by high membrane curvature, most notably the leading edge of the cell, which has a radius of curvature of 50 to 100 nanometer and intermediates in membrane transport including intracellular vesicles and endosomes. The motility process involves continuous reorganization of the morphology and curvature of these membrane domains. The spontaneous curvature of a membrane and its bending elasticity are determined by the composition of the membrane. The local curvature of the membrane is determined by these characteristics together with the forces acting on the membrane including cytoskeletal generated forces and pressure gradients across the membrane. Rather than being a passive consequence, local membrane curvature is dynamically determined by a continuous interplay between shape and composition. Curvature sensing lipids and proteins respond to membrane curvature and at the same time induce spontaneous curvature by energetically favoring curved morphology and by inducing changes in local cytoskeletal dynamics and first generation. The structure of lipids and membrane proteins and their interactions can favor membrane deformations, wedge-shaped lipids or proteins incorporated within the bilayer induce local curvature. The correlation between lipid composition and local curvature is clearly demonstrated by in vitro experiment in which giant unilamellar vesicles composed of several lipid species can spontaneously phase separate into domains which vary in both composition and local curvature. Furthermore, expression of membrane proteins, for example the Shiga toxin in cells, can induce the formation of highly curved membrane tubules in which the toxins is highly enriched. The proteins with domain that insert into the bilayer, for example an amphipathic alpha helix domain, can also sense and induce curvature. Scaffolding proteins that binds to the membrane provide yet another important mechanism for inducing curvature. One of the best characterized families of scaffolding proteins is the BR domain protein family. These proteins contain a rigid curved domain which interacts with membranes via hydrophobic and electrostatic interactions. The different BR domains proteins are characterized by different natural curvature. In particular, the FBR and NBR domain proteins induces positive curvature, whereas the IBR domain proteins induces negative curvature. In vitro experiments have shown that the BR domain proteins can spontaneously bind and deform membranes on their own, and that the type of structure formed depends on the protein structures. In general, the action of curvature favoring lipids and proteins and scaffolding proteins is viewed as being highly cooperative membrane deformation in cells typically involve a very large number of curvature sensing and curvature inducing molecules. The BR domain proteins affect membrane's curvature by other means also. Many BR domain proteins have additional domains, for example the SH3 domain, which binds and activates proteins from the WASP to WAVE family and thus promotes actin nucleation and polymerization. In vitro work has shown that activation of neutral Wiskott-Aldrich syndrome protein-mediated actin polymerization by proteins containing an FBR domain depends on membrane curvature. The actin polymerization enhancing activity of mixtures containing neutral Wiskott-Aldrich syndrome protein and FBR domain proteins was measured by use of pyranose. Addition of liposomes to the mixture significantly enhanced actin polymerization rates. Moreover, the actin polymerization rates were found to vary for liposome preparations that differed in their average size. These results directly suggest the possibility of feedback between curvature and actin dynamics. Curvature-inducing proteins stimulate actin polymerization in a curvature-dependent manner, and actin polymerization can generate forces which induce curvature. The leading edge of motile cells is characterized by a thin protruding region with high membrane curvature. This thin structure is maintained by restricting actin polymerization to the apex, and it is clear that localized pushing forces induced by growing actin filaments are important for generating and maintaining this high curvature. Although it is known that actin-related protein 2 by 3 which nucleates actin filaments is regulated by membrane-bound activators, primarily proteins from the Wiskott-Aldrich syndrome protein 2 wave family. It is still unclear why activation is concentrated only at the very leading edge. It is moreover unclear how the preferred directionality of the nucleated filaments towards the leading edge is established. In membrane transport and flow, the transport between internal membranes and the plasma membrane through endocytosis and exocytosis are essentially the only way for lipids and transmembrane proteins to enter or leave the plasma membrane. Endocytosis includes several different processes, including clathrin-mediated and capillary endocytosis, by which patches of the plasma membrane invaginate inward and bud off to form vesicles, thus reducing the area of the plasma membrane. Conversely, in exocytosis, intracellular vesicles fused with the plasma membrane so their lipid and membrane protein content is incorporated into the plasma membrane and their internal cargo is released to the extracellular environment. Together, these processes are of central importance in determining the composition of the membrane and its overall surface area. The membrane transport between internal membranes and the plasma membrane accompanies movement in all cell types. Furthermore, in some cases, there is evidence of polarized membrane trafficking in which either the rates or composition of vesicle trafficking vary along the front to rear axis of the cell. The overall rates of transport between internal membranes and the plasma membrane vary among different cell types. In Dictyostelium amoeba, it takes 3 to 10 minutes to replace an area equal to the cell surface area, whereas in fibroblasts, it takes approximately 1 hour. Rates of endocytosis will depend on the availability of internal membranes, which also depends on membrane biogenesis. Moreover, as discussed below, the rates of endocytosis and exocytosis depends on the tension in the membrane. Inhibition of membrane trafficking in several cell types, for example fibroblast, endothelial cells, and dictyostelium, reduces persistent migration and reduces cell speed. Although these perturbation experiments clearly implicate membrane transport in the motility process, the mechanisms and pathways involved are still debated. One of the main functions suggested for membrane trafficking is the recycling of membrane components such as cell adhesion molecules. Other possible functions including lipid transport to induce membrane flow to the leading edge and the establishment and maintenance of cell polarity. The importance of membrane transport to motility probably varies among different cell types, and research is needed to clarify its functions. Cell movement requires continuous recycling of essential membrane components. For example, membrane proteins such as integrins, which couple the actin cytoskeleton to the substrate, must be continuously replenished at the leading edge as the actin flows rearward in the cell frame of reference. A continuous flux of bound integrins move with it. To maintain motion, there must be a counterflux of adhesion molecules entering the leading edge. Recycling of transmembrane proteins, for example, integrins, can occur via several mechanisms. Within the bilayer, passive diffusion can contribute, but it is probably too slow to compensate for the retrograde flow of adhesion away from the leading edge. Thus, at least in rapidly moving cells, active transport is likely to be needed. Directed motion through the membrane can be driven by bulk membrane flow or motor-driven transport. Alternatively, membrane transport via polar membrane trafficking, in which integrin-containing vesicles are targeted preferentially to the leading edge, can provide means for directed transport. Alternatively, membrane transport via polar membrane trafficking, in which integrin-containing vesicles are targeted preferentially to the leading edge, can provide means for directed transport. Although integrin transport has been investigated to some extent, or recycled back to the leading edge is still largely unanswered. In most motile cells in which membrane dynamics have been directly measured either by single particle tracking of lipid-bound particles or by photobleaching or photoactivation, no net membrane flow has been observed. In this figure of membrane transport and recycling of adhesions, in letter A shows that adhesion complexes assemble in a hierarchical fashion from integrins embedded in the membrane and cytosolic proteins. Nascent adhesion form at the leading edge and attached to the substrate. The actin network growth and flow away from the leading edge leads to a rearward flux of adhesion. In letter B, where various possible mechanisms for recycling integrins back to the leading edge have been suggested, passive transport by diffusion will tend to equilibrate the integrin density but may not be fast enough for efficient recycling. Active transport to the leading edge can occur with the help of motor proteins, which drag the integrins within the membrane by drift induced by bulk membrane flow or by polarized membrane trafficking. Excess endocytosis reduces plasma membrane area, whereas surplus exocytosis increases it. The extent of change in surface area during motility varies among different cell types. During motility of Dictyostelium amoeba, substantial dynamic changes in surface area have been observed for individual cells over time, whereas rapidly moving fish keratocytes have essentially constant surface area as they move, despite changes in their overall morphology and speed. The function of surface area changes during motility is still unclear, whereas in Dictyostelium amoeba it seems that surface area regulation is an active part of the motility process. In keratocytes, membrane area remains fixed and the membrane seems to be stretched out around the cytoskeleton, imposing constraint on shape and movement. Rapid changes in cell surface area can also be mediated by budded and folded membrane domains that act as a membrane reservoir and enable cells to respond to sudden changes in membrane tension. To prove the membrane reservoir in motile fibroblasts, membrane tether were extracted from the cell membrane. The initial force required to pull tethers was found to be independent of tether length, indicating that the membrane flowing into the tether was taken from a reservoir. At some point, the tether force started increasing abruptly in an exponential fashion, indicating depletion of the reservoir. The extent of the membrane reservoir in fibroblasts is relatively small, 1%, but it could be larger in other cell types in which membrane folds and invasion action are more abundant. Recent work has shown that cavulae, which are cup-shaped membrane invagination, enable endothelial cell and muscle cells to undergo fast changes in cell surface area. Cavulae are actively generated through an adenosine triphosphate, an actin-dependent mechanism, and can then flatten out rapidly in response to cell stretching or swelling. This enables cells to buffer changes in membrane tension and prevent rupture of the plasma membrane. Overall, the questions how cells regulate their surface area and how changes in surface area relate to the motility process are still largely unanswered. Although a cell probably cannot directly measure its surface area, it is thought that cells respond to changes in membrane tension. Thank you for listening and stay safe, everyone.